Hello friends, welcome to all of you and welcome to the bio4u.com and in the playlist bioinformatics. Today I am going to explain a very important part of the drug design, that is the Lipinski filter. And this Lipinski filter is using to find out whether the molecule we collected, that is the ligand, is suitable for the drug design or not. If it follow 5 criteria, that is a Lipinski filter is forwarding 5 criteria. Out of the 5 criteria, if 2 criteria is following, then that is okay for the drug designing molecule. Okay, so that is where I am going to explain in the following video. Let us see the following video. A brief introduction to drug design. Design a novel drug is one of the biggest challenges faced by the pharmaceutical industry. The use of computer accelerates the process of drug design, which is a time-intensive process, and also reduces the cost of whole processes. Computational methods are used in various forms of drug discovery like QSAR, virtual screening, and structure-based drug designing methods. Among these, structure-based drug design is gaining importance due to rapid growth in structural data that is available in RCB and in nucleic acid data bank. This structural data can be used in molecular modeling to design lead molecules based on the structural features of the active site. Let us see how the drug designing is going on. And in this video, I am going to explain only a part of the drug design, that is the Lipinski rule, the 5 rules and how it can be applied. Okay, let us go. So now we are moving to the next part of the drug design, that we have to collect the ligand molecule or the drug molecule, okay, and the protein molecule. For that, we can go to the a tool that is called a Sanjeevani tool. So let us go to the Sanjeevani tool. So this is the Sanjeevani tool. In the Sanjeevani tool, there are two parts, that is the ligand and protein DNA metalloproteinase. So first we have to collect the ligand, that is a drug molecule. Okay, so just click here, you can see the NRDVSM database and a million molecule database, natural product database. Okay, if you click, I suggest you just to go to the NRDVSM database, because it is more convenient comparing to the million molecule database and a natural product database. So let us click on this point. So you lead to a new window. In this window you can see some of the default values, that is a molecular weight, it must be between 480 and 150, and a log p value, it must be between 6 and -1. Hydrogen bond donor must be between 0 to 3. Hydrogen bond acceptor must be between 2 to 9, and molecular refractivity must be between 40 and 130. So according to your requirement, you put these values and then click on the search, and it will lead to a new window. In that window you can see the many different types of the drug molecules. From that drug molecule you collect one and then you search the validity of that molecules, whether it falling in the drug criteria or not. For that we have to use another software, that is the Marvin Sketch tool. For that I will explain the details of the Marvin Sketch, how to use that and how can you find out the drug molecular criteria, etc. Okay, so let us go forward. After searching suitable drug molecule and a target protein, it should follow the protocol designed below. This is the protocol. So we already explained this one. So we collected the drug molecule here. Then after that we will collect the protein molecule and the drug molecules criteria, that should be fall in the 5 criteria. Then only we can use that molecule for the drug design. But our purpose of this video is not a drug design. So our purpose here, actually this video is explaining how to find out the criteria, whether a molecule is satisfying all the criteria or not, so that Marvin Sketch tool, how to use the Marvin Sketches tool, and how to find out the molecule is belong to the or satisfy all the criteria as a drug. Okay, so during the drug design, first we collect this molecule, then it will filter, and then uploading both the drug and the ligand and this protein molecule, then it will go docking purpose. And after the docking, it will give certain data, and from that we can identify the drug, or we can design the drug. That is another part of the video. So in this video, the next part I will explain how we can use the Marvin Sketch tool and find out whether the molecule is falling under the drug criteria or not. Okay, so that much. Now let us go forward. Now we can move to the Lipinski filter. So this Lipinski filter is using to find out whether the molecule or the ligand we already collected is satisfying the drug criteria or not. There are 5 drug criteria. The molecule must be satisfied with this drug criteria. Okay. So before proceeding the protocol for drug designing, one must check the validity of the drug molecule. All the molecule cannot be satisfied the criteria required for drug molecule. To check the validity, we commonly use the Lipinski filter. Okay. So this video explaining the details of the Lipinski filter methodology. Okay, so here Lipinski rules, there are 5 rules that are helping to distinguish whether the molecule is satisfying the drug criteria or not. Okay, so what are these 5 criteria? Out of the 5 criteria, 2 criteria must be follow the molecule, otherwise it cannot be satisfied as the drug. Okay, so what are these criteria? Molecular mass less than 500 Dalton. It must be less than, if more than 500 Dalton, that cannot be satisfied as the drug molecule. Okay, high lipophilicity expressed as a log P, that must be less than 5. Okay, less than 5 hydrogen bond donor must be less than 5. Similarly, hydrogen bond acceptor also must be less than 10, and molar refractivity should be between 40 and 130. If more than or less than that, it cannot accept as the drug molecule, and the mission will fail. Okay, so these filters help in early preclinical development and could help avoid costly late stage preclinical and clinical failure. So that much significance for this Lipinski filter. So if we are not using the Lipinski filter, we have to pay more amount or costly. It must be very costly after that. Okay, so failure to draw chemical structure, click here. That means we will go to the Marvin Sketch tool and follow the instructions given there. Then we can make a PDB file from the 2D structure, or from the 2D to 3D structure. 3D structure means that is a PDB file. Okay, so let us move forward. First, Lipinski filter. Click here this hyperlink. It will lead to the Lipinski filter. Okay, this is the Lipinski Rule 5 and the software. So here the choose the file. This file must be PDB file. Otherwise it cannot accept 3D. PDB file means it must be 3D file. So if you have the structure of the molecule or 2D file, this 2D file must be converted into PDB file by using the Marvin Sketch tool. So that will explain later, the Marvin Sketch. Okay. And first put here the molecule, then select the pH value. Usually pH value is in the neutral value, that is a 7. Then here submit. So first select a PDB file that already I prepared. Okay, so this is the one of the PDB file. So collect that, then submit here. It will lead to a new window. So here you can see the result. This is the result. The mass is 458.0. That means it is below 500. Okay. Hydrogen bond donor 1. That is below 5. It is okay. Hydrogen bond acceptor 8. That is below 10. That is okay. Log P value 4.35. That is below 5. That is okay. Molar refractivity 120, that is below 130, so that is okay. So all the 5 criteria is followed. The molecule I already selected. That means it is a very good ligand or very good drug molecule. So because it is following all the 5, at least 2 criteria must be followed. Okay, so that is the Lipinski Rule 5. Then let us go forward. So this is the Marvin Sketch input tool. So you have to put here Lipinski tool, not a Marvin Sketch. So here you have to put the PDB file, then select the pH value, commonly is 7, neutral, then submit here. So I already explained this point in detail. Okay, so input file should be in the following format. It must be in the PDB mol2 XYZ SDF or SMI. So commonly we using the PDB file. Okay, the input file name should not containing white space. No white space is requiring, or it must be avoided white space. Browse and upload the file from the computer and click on the submit button. So this submit button, and that will submit. And if the result were not showing, please recheck your input file. Something problem in the input file. If the input file is one of this, the result must be there. Okay, then draw the chemical structure. Click here for that the Chem Sketch or the Marvin Sketch tool will open, and follow the instructions given there already. I will explain. So that is the this one. So let us move forward. So this is the Marvin Sketch tool. How to use the Marvin Sketch tool? First, download the Marvin Sketch tool from the site given below. So this is the Marvin Sketch tool downloading site. After that, you have to verify that the tool whether working properly or not. So how to download that? Let us go forward. So this is the hyperlink. Okay, double click on the hyperlink. You can lead to the site. So this is the Marvin Sketch site. In that site, you scroll down, you can see here Chem Axon. So first download this Chem Axon. After downloading the Chem Axon, you can see that in the Chem Axon window, you can see that Marvin Sketch tool. So you download not a Chem Axon download, you download the Marvin Sketch tool download. After downloading the Marvin Sketch, you have to install that. Then also it cannot work, because it requiring the license file. So to download the license file, here is the option here, download the license file. And after downloading the license file, you just open that zip file. Then after extracting that, you have to install that license file. After installing the license file, the Marvin Sketch tool can work smoothly. Okay, so after the installation of the file and the Marvin Sketch, you can use that very smoothly. So how can it appearing that this is the Marvin Sketch tool? In this Marvin Sketch tool, you have to give a structural formula. After giving the structural formula, you have to convert that into PDB structure. So how it can convert into PDB structure? So I will collect the Marvin Sketch tool from the window. Okay. So this is the Marvin Sketch window. So this is the Marvin Sketch. Okay. So in this Marvin Sketch window, you have to upload the 2D file or the structure. Okay. So just open that. So here I already downloaded a PDB 2D structure. So this is the 2D structure. Open that. Okay. So this is the 2D structure. After open the 2D structure, you just to go to the structure option. In the structure option, add option. In the add option, you just click the explicit hydrogen. Okay. Now the hydrogen is added. Explicit hydrogen is added here. Then after that, again go to the structure. Then here click on the clean 3D structure. Okay. So after clicking on the clean 3D structure, you will get a PDB structure. This is the PDB structure, and this PDB structure you have to save. Okay, so this is the PDB structure. See here, this is the protein data bank PDB structure that is saved on the desktop. Then later that you have to use power. It already first downloaded that you have to use for the Lipinski criteria, checking the Lipinski criteria. Okay, so that much. Then we move to the next part of this video. Hello, this is the Marvin Sketch tool drawing chemical structure, the procedure. Okay, so as per the procedure, first draw the structure, and already explained all these points. Add explicit hydrogen structure, add explicit hydrogen, then convert the structure from 2D to 3D structure. For that purpose, you go again to the structure, then click on the clean 3D, then clean in 3D, then you will get the PDB structure. Then save the PDB format on your desktop, upload the PDB file in the Lipinski drug file filter, and you will get the result within a fraction of a second. So that much in this video. So I already explained how the Marvin Sketch tool you can use, and how the drug molecule can be analyzed for its criteria, 5-point criteria, that is Lipinski criteria. If that follow at least 2 criteria, then that can be used as a drug molecule. So that will save the time and expense after the preclinical trial. So that is a very good idea for that. So I request all of you, please subscribe this channel and like it. Okay, thank you very much.